Good morning, everyone. My name is Enrico Santus, and I am a senior data scientist at Bayer. I'd like to thank the organizers for inviting me today to this brilliant event. Today, I will present a project that was not done with Bayer, but instead with the University of Udine and the Hong Kong Polytechnic University. And it is about analyzing vaccine opinion on Twitter. The work that I will present in the next slides was possible only thanks to this group of students that are reporting in this slide. They are led by Professor Giuseppe Serra, myself, and Emanuele Chersoni from the Hong Kong University. So our goal was to create a portal that could analyze the COVID-19 vaccine opinion in social media. We decided to start with Twitter because it was the easiest source of information we could analyze. And our goal was to get insights that could have been useful for the public, for journalists, for pharmaceutical industry, and the decision makers. The portal is reachable at the address you can see in the bottom left of the slide. And it works as I'm going to illustrate to you right now. So you will see here that there are several modules. The system is basically collecting everyday tweets from Twitter indeed, that contain keywords or hashtags that are relevant to vaccine and COVID-19. Then there are several modules that check the location of the tweets, the hashtags that are mentioned, and check the correlation between hashtags, the URLs that are mentioned into the tweets. And so we can basically analyze the news that are commented and the sentiment of the tweets themselves. And finally, the most important module of this system, which is the adverse event detection. And I will spend my presentation on this specific module. So let's look very quickly at the localization module. As you can see, there is a map. And the darker the countries, the more tweets are coming from those countries. You shouldn't be surprised that the English-speaking countries are darker than others because we are focusing on English tweets. And here you see the list of the most utilized links in the tweet for a specific day, the day I made the slides. A good news is that most of them are reliable sources such as USA Today, The Guardian, AstraZeneca, and so on. So we can be happy that the people actually mostly comment on reliable source of information. And finally, we get to the most relevant module where I get into details, and I hope it will raise your interest. So every year, as you probably know, dozens of drugs are released in the market. After that, the pharmaceutical companies test their safety and effectiveness in clinical trials. However, clinical trials are unable to identify all the potential adverse events. For example, they are unable to identify all the potential drug-to-drug interactions, as the clinical trials, anyway, are done on a small amount of people, relatively small amount. For this reason, pharmacovigilance needs to surveil the market to timely capture adverse events and minimize their harm. One way of doing so is to rely on the spontaneous reporting systems that are those systems where both doctors and patients can somehow report the adverse events. For example, one of the top ones is the FDA Adverse Reporting System, also known as FAERS, which has received until 2018, it has received over 16 million of adverse events, 9 million of which were classified as serious, 1.5 million of which were related to the death of the patient. So the increment of reports between 2007 and 2017 was of about 400%. Despite so, it is estimated that over 95% of the adverse events are not reported. And this is a very serious problem, if you think about it, because they are responsible for somewhere between 5 and 8% of the hospitalizations. In recent years, a growing number of patients is discussing their adverse events in social media and other health forums and other outlets that can be found online. Of course, controlling those media is very complicated because they are subject to the 3 V's of the big data, meaning velocity, volume, and variety. So the automation of the monitoring of those media is crucial, and the NLP community has been somehow called in to utilize automatic systems for the identification of adverse events. In recent years, many workshops and shared tasks have been organized by the NLP community in order to address this challenge. And of course, this challenge is harder than you can imagine. First of all, because the majority of tweets that mention a drug or mention a symptom are actually not always speaking about an adverse event. So we don't want to have a false positive, right? The other issue is that the tweets and anyway the forums and so on utilize a lot of informal language such as slang, metaphors, and so on. And as you probably know, natural language processing is stronger when the language is literal. And finally, the typologies, as we said before, the variance of text typologies is very high. You may have very short and noisy text in Twitter, or you may have long and structured text in forums. So in order to start our project, we focused on one of the most famous shared tasks on adverse event detection, which is the SMMM4H shared task that is run every year since 2016. SMMM4H stands for Social Media Mining for Health Applications. And the main goal of this task is to identify which tweets contain an adverse event. And once you know that there is an adverse event in them, you want to identify which words are the adverse event. And to do so, you need to basically classify each word on whether it belongs to the adverse event or not. As you can see from these pictures, where head hearts is the part that belongs to the adverse event. If you look at the systems that were submitted to the shared task, the best performing ones were complicated ensemble methods of BERT models. You know, BERT is a very powerful transformer that has been utilized for many goals, and adverse event extraction was one of those. When we enter in this challenge, our goal was to create a very simple architecture, utilize possibly something that was already pre-trained without any big personalization, and also to make it work across different type of text typologies. Finally, to try to achieve this goal, since the most well-performing system were based on BERT, we wanted to look at the various BERT variants and try to see which ones was actually the best candidate. And we found this into SpanBERT. SpanBERT is an optimized BERT architecture for predicting spans of text rather than single words. And you can see this in the picture below, where an American football game has been masked. And this full span has been predicted based on the information in E4 and E9. So the words just out of the boundary of the span. And through this system, through this training approach, the system has to learn a lot of contextual information that you will see it will be necessary later on when we talk about adverse event. And so the question is, why is it useful in our case? Well, first of all, because adverse events are actually spans. You rarely find only the word headache. You generally find something like my head heart or something like that. On top of this, adverse events are generally expressed with idiomatic expressions, which contains multiple words, and the meaning of every single word in idiomatic expressions is basically known. The meaning comes from the full idiomatic expression. So you don't want to capture the meaning of the single word, but you want to capture the meaning of the span. Another important thing is that when you deal with spans and you know that adverse events are reported in these ways, you end up having a system that is more cross-genre flexible. So this is the reason why we decided to adopt SpanBERT. And here there is a presentation of our architecture. As you can see, we get the text in input in the bottom of this image. text is annotated like in the data set is annotated with IOB labels. So we get a text in input, then the spam BERT returns some embeddings for each word. These embeddings go through a linear layer and the softmax, which return the label. This label is temporary because this label is passed then to a CRF module. A CRF module is nothing more than a statistical system that learns the transition between the various labels. And it knows that, for example, you may not have a BB like you see in the temporary labels, and it turns it into BI. Okay, that's the final layer that returns the output labels. In order to test our system, we wanted to verify its performance on SMMM4H, of course, because we focused on that, but also on a different typology of text that were in CADEX. So SMMM4H, we said it contains over 2,000 tweets, which are short and informal, while CADEX contains over 1,000 forum posts, which are longer and more formal, and they contain also more medical terms. We created several baselines for as a competitors. One of them is BERT, of course, then BERT plus CRF, then spam BERT without the CRF, and then finally the top 4 performing system in SMMM4H 2019. In the slide, you see the results, and you may notice that the spam BERT plus CRF actually outperforms all the other systems, actually even by significant values in most of the cases. You can notice also that the results on CADEC are much higher than the results on SMMM4H, and the reason is because of the typology of the text, the informality of Twitter. The thing that we notice also by looking at the spam BERT output is that it generally extracts the full idiomatic expression, while all the other systems were extracting only some words of the adverse events. So it's more comprehensive. We submitted our system to the formal leaderboard of the shared task, and we ranked first. We are currently at the top of it. And this, again, possible to the weight of the students we are collaborating with. The follow-up paper describing all our approach, the error analysis, and so on was submitted at the workshop for health intelligence in 2021, and it got the best IBM short paper. Once you see that this system works, then we have to create an interface for our portal. So we were talking about the portal with the map. You remember the map, the links, and so on. And then the next one is this word cloud, which contains all the possible adverse events related to the various vaccines. In this case, Pfizer, AstraZeneca, Moderna. You have read the news very recently, I'm pretty sure, and this news we're talking about adverse events from AstraZeneca. And our system actually, it's interesting that it kind of identified those adverse events pretty early in these days. So we are very proud that this system may be actually helpful to monitor the adverse events in the future. And here you see, if you click on one of those adverse events, it opens all the tweets that are related to that specific adverse event. You can read them, you can download them, and also you can check whether they are positive or negative. In this case, they are the green ones are positive, the red ones are negative. This is another useful feature for those who want to analyze better the tweets. And another information that we provide is this one. We basically give the correlations between the adverse events and the single vaccines. And you may notice, for example, that allergic reaction are much more correlated with Pfizer and Moderna, while headache is much more correlated with AstraZeneca. To conclude, I presented our portal. Our portal is now working, is getting tweets in input and giving you the adverse event about the vaccines, the COVID-19 vaccines. But actually, it's a very flexible framework that can be adapted to any drug in the market, and it may also be adapted to any input type. For example, we may work with pharmaceutical companies and get the information from the call center, identify the adverse events in the call centers, or from the chatbots, and so on. And this may help to help these companies to monitor the market. That's all from my side. If you want to participate to the Q&A, I'm happy to answer. And also, these are my references if you want to look at my website and my email. Thanks a lot to the organizer. Thanks a lot to you for your attention. And I'm looking forward to the Q&A.